This is examining developmental genetics with the hypothetical spike pathway of the spiny beetle. The goal of this video is to help understand the logic of gene-to-gene repression and activation and how mutations in a developmental sequence can alter the outcome of the pathway. To do this, we will be examining a hypothetical pathway that determines spike formation in the ectoderm cells of the spiny beetle. So here we have our pathway of spike formation in the spiny beetles. So we'll go over it real quick. The S gene activates the P gene, which also stimulates the I gene. That's what these arrows stand for right here. I have them highlighted in yellow. Now the I gene represses the K gene, which in turn represses the E gene. So the fact that we have the I gene repressing the K gene means that this E will be available to produce our spikes. And this is the normal developmental pathway. So here we have our first mutant strain for our developmental pathway network, and we are going to be insufficient or mutant for the P gene here. So I've gone ahead and crossed that out for us. So without the P gene, we are not able to produce the I gene. And this I is important because it is inhibiting the production of K, which inhibits E. And E is what gives us the spikes in the ectoderm cell. So without P, we are not able to use our I gene to produce the I gene to inhibit K. So with K present to inhibit E, we will not produce any spikes in this particular scenario. So here is our second mutated strand. In this particular scenario, the I gene will be the mutant, so we will not produce a functional I gene here. And this is actually will have a similar fate as our previous example. So without this I gene, we are not able to repress this K gene right here. And without the repression of that K gene, we are not going to be able to produce E, which is going to lead to our final outcome of spikes in the ectoderm cells. So in this particular example, with no I, we will not produce spikes. No spikes. Okay, so for our third mutant strain, we will be mutant for I and E, meaning we will not have a functional I or E gene down here. And this one's actually pretty easy to solve for. So because we do not have this last part of the developmental network, the E, before we get to our spikes, we will not be able to produce spikes in this ectoderm cell. There'll be no spikes because this E is missing. So regardless really what this I is doing, we're not going to be able to see spike formation. Our fourth and final mutant strain is going to be mutant for S and K. I've gone ahead and scribbled them out. And so while we won't be able to produce P or I to inhibit K, K won't actually be present to inhibit E. So we will be able to produce functional E genes that aren't inhibited by K, so we will be able to produce spikes. So yes, spikes will be produced in our fourth mutation scenario. My hypothesis for why not all cells in the ectoderm have spikes is because the variability increases the individual fitness of the spiny beetle. Beetles survive selection because of some spike expression for optimal protection and functionality. And this is built in molecularly to these pathways, so that not each ectoderm cell will express spikes, but at least some will.